what happens is cells that are under stress or deprived of nutrients can create a double membrane and non-specifically engulf cytoplasmic contents, and then they degrade them by fusing the resultant double membrane vesicle with a lysosome, and that generates nutrients which can be used for cell survival in times of stress or starvation. And it's very conveniently monitored by using a GFP-tagged LC3, which stands for light chain 3, which is an important component of the autophagy pathway. And you can see that when, oh, well, there is a laser pointer that works, hopefully. There you go. That you can see that when the cells are happy in full media, this GFP is diffuse, and when the cells are starved, you get these dots which correspond to autophagosomes. This is very widely used in the autophagy field. Now, the reason that LC3 is such a good marker for autophagy is that it actually becomes covalently lipidated and inserted into the autophagosomal membrane. And as you might expect, there's a very complicated sort of upstream regulatory pathway here, and there's a kind of E1, E2, E3-like, almost ubiquitin-like pathway going on here, and indeed LC3 is structurally related to ubiquitin. But the point about it is a very good and effective marker for autophagosomes. So why do we care about autophagy? It is important for host-pathogen interactions. And what you can see here is the way in which it's been adapted to restrict, for example, salmonella. So when salmonella comes into the cell, it's detected by adapter molecules on the basis of a burst endosome and on the basis of ubiquitin, which decorates the invading bacterium. And it can be dragged into these autophagosomes and restricted. And the molecular details of how that works are beginning to be sort of worked out. And we know that some viruses are also restricted, probably not by this pathway. But most viruses seem not just to evade autophagy, they actually seem to benefit from autophagy. And you can imagine that conceptually, if you're a pathogen and you stimulate autophagy, you too could benefit from the nutrients that autophagy affords, so long as you can evade degradation. Now, it turns out that influenza virus is very much in this sort of side of the equation here. Autophagy seems to be working in favour of influenza, and influenza seems to be deliberately affecting autophagy. So this is how we're used to thinking about influenza, a sort of round blob here with the hemagglutinin and neuraminidase and a much less numerically sort of large amount of M2 in the membrane of the virion. But it's important to note that most pathogenic isolates of influenza are actually filamentous, so they produce these enormous long filaments. And you can imagine that this requires a vast amount of membrane, vast amount of protein. And so you can imagine that in principle it would be a very good idea for influenza to be able to stimulate a catabolic pathway to give it more nutrients to make more virus. So we did a very naive experiment. People have had a look at this before, but we discovered something just a little bit new. If you take GFP LC3 cells and you infect them with influenza, you do find these dots corresponding to autophagosomes, but you also find this very strange phenomenon where you get GFP LC3 actually at what appears to be the plasma membrane. So that's not where LC3 ought to be. LC3 ought to be decorating autophagosomes, and it appears that what you've got here is autophagosome-derived membrane actually at the plasma membrane. And as per a previous report, this depends on the influenza M2 protein. Because if you take a delta M2 virus, you still get very few autophagosomes, not much to speak of. Essentially, it's much more similar to the mock than it is to the actually infected cells. And this is not some sort of bystander phenomenon. So if you infect cells at a low multiplicity of infection, you find that just these 3 cells have been infected here, as evidenced by M2 immunoreactivity. You can see it's just these 3 cells that have redistributed their GFP-LC3 to these kind of large accumulations of what are probably perinuclear autophagosomes here, but also to the plasma membrane. And there's this very suspicious colocalization with the M2 protein. And this is just to show you that this is sort of all really true, if you see what I mean. The delta M2 does not relocalize. And if you take an LC3 that can't be lipidated and therefore can't be inserted into a membrane, it remains diffuse in the cytosol. So it seems to be dependent on autophagy, and it's dependent on M2. And it really is the plasma membrane. So this is a mock infected cell. You can see the occasional autophagosome here. And this is an infected cell. You can see little virions budding off here. And you can see quite a lot of, this is an immunogold staining here, obviously, on electron microscopy. You can see that it's actually at the plasma membrane. And if you take the version of LC3 that can't be lipidated, it just remains diffuse in the cytosol. So here's another wild-type virus infected cell, and you can see quite a lot of the GFP actually at the plasma membrane. And it really does produce little autophagosomes too. You can see these pretty little double membrane structures which are also labelled. So what about this very suspicious colocalization with M2? And what is M2? So you probably know that this is a proton channel. The structure is fairly well known except for the last sort of 30 or so amino acids. It's a 97 amino acid tetrameric proton channel, quite sort of deeply embedded in the membrane. And we know that it's got this main function of enabling the virus to sort of uncoat, as it were, to disassemble when it's come into an acidified endosome. We also know that it's got a perhaps rather less well-defined role in assembly and budding. It's known that there's an amphipathic helix here, which probably mediates some sort of escort-independent membrane scission. But even further downstream, sort of further down the protein, we find something which might be interesting for those of us who are interested in autophagy. So what we find is something which looks as though it could be a Lear motif. That's an LC3 interacting region, which, as the name would suggest, binds to LC3. And the mechanism whereby that occurs is you form a beta strand in your Lear protein, and that interacts with a beta strand in the LC3 to form an intermolecular beta sheet. And if you look right at the sort of C-terminus of influenza M2, that's amino acid 97, which is the last amino acid, you find this motif which is predicted to be a beta strand, and it's got this FVSI motif, which is very similar to Lear motifs you find in human proteins such as ATG13 and octonirin. And it's also surrounded by these acidic residues, which is another typical characteristic of a Lear motif. So the bulky sort of hydrophobic residue here in position 1 is very important, as is this isoleucine or leucine in position 4. So does it bind? The answer is yes, it does. It's very strongly conserved. I don't know if you can make it out. This is the wild type here. This is if you've chopped off all of this region. And if you mutate the hydrophobic residues, you abolish binding to LC3. The motif, like I say, is very well conserved. You can have either an S or an N in this position, depending on the isolate of influenza you're talking about. And if you make that substitution, the S that we have in our laboratory PR8 strain to N, you actually preserve, possibly even enhance binding. So the binding's preserved. And we can calculate a KD by purifying the protein and getting a labeled peptide by fluorescence anisotropy. And the KD is about 10 micromolar. And we can pull it down in infected cells. But if we make a mutation in the segment 7 so that this F is now an S, and that hydrophobic residue is therefore gone, and therefore the Lear motif is no longer operational, you fail to pull down M2. So it is a Lear motif. So what happens when you make a virus which doesn't have the residues in the Lear motif? Well, you still do. So this is the wild type. That's, if you like, the alternative wild type. You can see plasma membrane localization. You can see plenty of these autophagosomes. If you have got rid of these hydrophobic residues, which are important for binding, you still do see some autophagosomes, but what you don't really see is very much at the plasma membrane. And this is sort of quantified down here. So you could imagine that there's a sort of very complicated regulatory pathways up here. You would predict that if all this is true, if you got rid of this ATG16, ATG5, ATG12 complex, the whole phenomenon would go away. And we don't know if it's going to depend on the canonical sort of stimulatory pathway via mTOR and so on. So we had a look at some of those factors upstream. And essentially, if you take up FIP200 by siRNA, this is FIP200 in the part of this ALK1 complex, quite upstream, if you like, in the signaling pathway, then actually very little happens. You still get plasma membrane localization, you still get autophagosomes. If you take out ATG16, which is, if you like, a core component of autophagy, then you don't see any redistribution. So it all seems to check out. Now, that's all very well, sort of pretty pictures and what have you, but does the virus actually care about this? And for a long time, we were a bit depressed because we thought we'd discovered something which was pretty pictures, but the virus really did not care about. Because if you put these mutations into the virus, so this is the I94S or F91S here, you get exactly the same titer out in cell culture. And I haven't shown it here, but the pathogenicity of these viruses compared to wild type also seemed to be on a very limited experiment in mice. It seemed to be pretty much the same. But it turns out that the stability of the virus is significantly affected. So PR8 is a spherical virus. It's our laboratory strain. And if you make these mutations, the viral stability is significantly affected. It drops off for about a log fold after a couple of days. If you take a filamentous virus, so this is where you've replaced segment 7 of the PR8 strain with the UDORN strain, and therefore you make the virus filamentous, the phenomenon seems to be a little bit more pronounced. But you broadly speaking, you're dropping about a log fold in infectivity after a couple of days of just being left out on the bench. It's a kind of lazy person's experiment. So we do think this is going to be biologically important. And filament formation is affected. So this is in the mud strain, which is filamentous. You can see these great big sort of strong cables of virus coming out here. Plenty of them in the S93, alternative wild type as well. In the F91S and I94, you still do form a few filaments, but they're kind of thin and a bit weedy and rather disorganized. And so this is sort of showing the same thing, looking at bundles of filaments in a kind of lawn of cells by immunofluorescence. So here's the wild type. Here you're looking down on it. Here's the same picture sideways on. This is like the Chicago skyline with all the skyscrapers up here. And this F91S, you still see a few filaments. It's a bit more like where I come from, Cambridge. It's basically flat. And this is to show that you see more or less the same phenomenon if you knock down ATG16. So you still will form the occasional filament, but far fewer than in the control. So this is more or less where we're at with this project. I've started my own laboratory back in April, and we're working on trying to decipher what this all means, how it is that this interaction somehow promotes filamentous budding, how it is that this interaction is important for stability. We've looked at things like lipid composition of the virions. We're going to have a look at protein composition by mass spec as well. At the moment, we don't understand exactly why the virions are unstable.